Okay, welcome everyone. Today I'm very honored to have Professor Pedro Mendes from the University of Connecticut Health Center. With a PhD from the University of Wales, Professor Mendes is a leading figure in computational system biology. He is very well known for developing influential simulation software like a COPASI and has contributed extensively to the field with research spanning from biochemical pathway modeling to reverse engineering. So please join me welcoming Professor Mendes, who will share his valuable insight into the computational modeling for biological system.
Well, thank you very much. Thank you for the introduction and for inviting me to this colloquium series. I'm very happy to give you a presentation. I'm going to talk about some work that I have been carrying out with my colleague Aaron Agmon, who was a new assistant professor that joined our center a couple of years ago already. Aaron was previously in the University of Stanford working with Marcus Covert on the whole cell E. coli model. And he has done a lot of work on various computational systems before. He's a computer scientist, but also a biologist. And I guess it's similar with me. So we've been joining together, trying to combine our software COPASI and Vivarium, or see how it couples. And I'm going to tell you about one application that where we're using both software. Essentially, we're studying how hybrid multiscale models that are based on differential equations and agent-based approach, how can we study the error, the approximation error that is introduced by that hybrid simulation method. So essentially, I'm going to just give you a brief overview of multiscale simulation and how people understand it and how it is most popularly processed in biology at the moment. I'll tell you a bit about the software framework we're using, and then we'll describe a benchmark model that we've been applying this to. It's actually a famous model from systems biology. And there's a little tale about that too, but I will be quick about it because it's not the main topic. And then finally, I'll be using that benchmark model and comparing full ODE models with coupled ODE equations, fully coupled, and then hybrid approaches where you use agent-based models that have in them smaller ODE models that then are coupled through the agent-based approach. Okay, so the first thing, as Jai mentioned, I've spent a lot of years developing COPASI. This is software for modeling by chemical networks and chemical networks as well. I have to say it's quite used in chemistry. To our surprise, we never publicize the software in the chemistry field, but it is about 50% of the papers published with COPASI are actually coming from chemistry for people doing simulation of what they call microkinetic models. So COPASI is essentially it's not a spatial software. It's only time dependent. It can simulate models based on differential equations, on stochastic differential equations, and also on the Gillespie stochastic simulation algorithm and their derivatives of that one. It allows adding discrete events and there's a large number of methods for analyzing the model, including, as someone mentioned before, including timescale separation analysis, for example. And also it's quite commonly used for parameter estimation. It's one of the features that it is more popular, but it does a number of other approaches. Importantly for this talk, I'm going to mention most of the work was done with a interface that we have developed where we can use COPASI directly from Python. And there's another one also for R, for the R language, but I will not be talking about the R version, but it's very similar to the Python one. Essentially, all the code runs in COPASI, in the binary compiled COPASI, but you can interface it through Python, and that's really how this whole work is based. So I'll talk a little bit more about that. So It's by now quite a bit popular software. We have about 100, just over 100 papers per year. It changes from year to year.
Mm-hmm.
2019 we were going up, but then I think COVID happened and there was a little drop after that.
Uh-huh.
Hopefully it will go up, but these are essentially a lot of publications use COPASI, about 100 per year at least, and all around the world as well. I did not zoom in here, but if you go to our website, to this webpage here, you can actually zoom in and see who in Korea also uses it. There's not a lot. In Korea, there's maybe 5 or 6 only dots over there. There's a lot more in the US and Europe. But as you can see, all the way from above the polar circle down to Tasmania, and east and west. And a number of journals where papers are published. As I said, to our surprise, Journal of American Chemical Society is #2 on the list,
Mm-hmm.
even ahead of PLOS Computational Biology. Okay, so COPASI is widely used for modeling time-dependent networks mostly. I mean, you can define compartments and you can define different topologies of compartments, but essentially there is no diffusion, there is no explicit space in these models. Now, in biology, for now quite a number of years, at least 2 or 3 decades, people have been talking about multiscale modeling a lot and practicing multiscale modeling. And the reason for this is that biological processes cross a very large range of time and spatial scales. This figure here is from a review that I wrote about 14 years ago or so. And you can see the spatial scale goes all the way from macromolecules to entire organisms. In fact, we could even go to ecosystems. There's even some papers where COPASI has been applied for ecosystems as well. And more importantly, even in time scales, things are even deeper. So you go from enzyme catalysis, which is on the microscale, the microsecond order. Of course, we could even go even lower if we think about quantum transitions in photosynthesis, but I don't want to get to that low. But let's say enzyme catalysis, which is widespread, microseconds, all the way to aging, which happens on the scale of 10 to the 9 seconds, which is about the lifetime of a human. So everywhere in the middle here, you'll see some of the processes that you just mentioned before, like circadian rhythms, a little bit on the large scale, but still not close to aging, of course, and quite far away from metabolism, which is at a very different scale. So coupling these scales can be an issue. Sometimes it's a benefit. If you're really focusing only on one scale, you don't maybe necessarily have to study the other scales. If you're thinking about hormonal regulation, it may be that you don't have to represent the mechanisms of enzyme catalysis in detail. And also you don't have to simulate the whole lifetime of the organism. So you can consider this process as being fixed because it's not moving fast enough, and this one is in equilibrium because it's sufficiently fast.
Mm-hmm.
However, if you want to couple this, and there's many phenomena where you have to couple them, people have been trying to address that, and multiscale simulation seems to be the natural approach. And in this approach can be done in different ways. Two of the most popular ones are the following. One of them, you separate the scales with different simulation methods, and then you couple them by some framework. So this is actually going to be what we're going to be talking about for the most of the talk. There is, however, another approach quite used in physics, but it could be also used in biology, so-called mean field approach, where some of the scales are represented through approximations, and therefore, in the end, you can combine them through that way. A very typical method in systems biology is to use represent cells as single agents, agent-based models, where each agent is a single cell, and then connect them at certain times. So the cells can, by and large, operate independently for a while, and then at certain times they're connected and there's communication. And that's the method that we're going to talk most about. So this is the summary of that approach. So it's multiscale simulation through agent-based coupled with ODE modeling, as we'll see later, although it doesn't have to be coupled to ODE models. So this slide is still a little bit more generic. So these agents are governed by rules or equations, and they're autonomous. So each agent can decide what its next behavior is going to be, just from its own internal variables and some environmental variables. And these agent-based simulations are actually quite important when you have cells or other elements that move locations. So if they're mobile and they move, and therefore they could have contacts with different cells at different times, that is not very amenable to representing that with sets of ODEs. You could do it, but it would be really cumbersome. Also, if the number of cells is variable, for example, you can have cells that are dying and cells that are dividing and forming new cells. So in that method, the number of cells is also not fixed. Therefore, you would have to add or remove differential equations to the system, and so that's also not very easy to do. And a final benefit, which is often highly seen, and we'll look at that as well here, which is that this methodology has the potential to be parallelized easily. So if you think about each agent being at least running for a certain amount of time independently from the others, you could dedicate them each to each different computer. And so for a while, all the computers could be running in parallel. But then, of course, they have to communicate at a certain point. So COPASI, even though not being a multiscale simulation approach, it's an ODE or stochastic simulation approach, has, however, been used in the past, coupled with other frameworks to implement models of the kind that I'm describing. So here I'm listing a number of them. This is not an exhaustive list. There's others that I haven't listed here, but these are at least some of the perhaps more popular ones. And the ones that have the COPASI symbol in front have already been coupled with COPASI. So, and I'll show you 3 examples with Computational 3D, Flame, and Repass. These are different environments, and people have used COPASI as the part that runs the ODEs. Okay. So first one, this is a paper from way back 2010, so a number of years ago already, where they simulated a human epidermis. So here the number of cells is variable, as you could see by different snapshots. Also, the states of the cells are very different. They color them by different types of keratinocytes, and I think they're labeled according to the content of keratin and how that evolves. They use this Flame environment, which is an agent-based approach with an interface based on XML, and they use COPASI to be carrying out the differential equation. So each of these cells that you see here, each of these circles, is an independent agent, and internally has a set of differential equations, which is this part here, the subcellular layer. In this case, these were models of the TGF-beta1 signaling pathway. And then the agent-based approach, the agent-based framework, decides when new cells have to be formed and keeps track of their locations and therefore their neighbors. So moving on to another different approach. This one was carried out with another framework called Repass, which is I believe a Java-based environment. And in this case, they modeled a mucosal immune responses. So they're simulating a portion of a mucosal tissue, which contains a very large number of cells of different types, as you can see here, various types of immune cells, but also of bacteria. You'll see the small dots are bacteria, and the larger colorings are the different cell types. And then these cell types, in this case, there are different signaling networks that each of them carry depending on what type of cells they are. Again, COPASI was used to run the internal biochemical networks of each of the separate and different cells. Again, this was still about 10 years ago or so. And then another one, the final one that I'll present, this was run with Computational 3D, which is a framework created by Jim Glazier at Indiana University. And in this case, there's actually 3 layers, and COPASI was used in 2 of them, the 2 extremes. The very high level, which is the human model. This is a pharmacokinetic, pharmacodynamic model. And at the lower scale, the metabolic model, where the drugs are metabolized. Intermediate layer is run with this Computational 3D, and they simulate a sinusoid structure of the liver, and then from there they scale it to the size of the whole liver. So in this case, you have hepatocytes, which are these green squares, and you have red blood cells going in a venous flow and also an arterial flow. So this model includes hemodynamics, so it includes the fluid dynamics of the blood in the sinusoid, and the processing and transport by red blood cells and hepatocytes and the serum as well. And they have a whole PKPD model in one layer, and then another layer, whole metabolic model for each one of these cells. And so COPASI was in this case used at two ends. So these are 3 examples. I don't want to describe them in detail. This is just to say that this is not new. People have been doing this throughout the years, at least the last couple of decades. So one of the things that I have always been puzzled about these approaches is the question of how much approximation, how much error through the approximation that is introduced by coupling the 2 approaches. So you have ODEs being run for a certain amount of time. Then there's communication, that is, values are exchanged between the different ODEs, and then they run again independently. And if you think about it, in reality, real cells, there's no such schedule of communication. It happens continuously all the time. And so there must be some approximation error that is introduced by this approach. However, as much as I have looked for, I have not seen anyone studying what that approximation error is and how could you control it. So that's what we're going to try to do here. And we're going to use a model which is amenable to be run with full sets of coupled ODEs because the cells are fixed. They are not, their number is not changing, and their location is not changing, and therefore we could actually represent them with sets of ODEs. As you will see, they're large sets of ODEs, but it is possible to be done, and in fact it was done also quite a number of years ago. And then we will also simulate that with the agent-based coupled to ODEs approach. And now we're going to be using Vivarium software, which my colleague Aaron Agmon has been developing. And I'll talk more about that in a second as well. So first of all, I'm going to tell you about the model we used for benchmark, which is the segment polarity network of Drosophila. This is actually a very famous model. It was published in the journal Nature in the year 2000. It has been cited many times. I think it has around 2,500 citations, something of that order. And it is a paper, the whole paper is about robustness. It's basically, their message in this paper was that there is robustness in the signaling network such that you get this segmentation pattern forming in the Drosophila embryo. These segments that are formed in the embryo are basically patterns of gene expression. If you tag fluorescent tags to certain proteins of this signaling network, you will see that they form patterns like that, these segments. And these segments are going to develop in different segments of the body of these insects. This is specific to insect development, of course, but it happens in similar types of things happen in other development of other embryos. So in this case, they represent each cell with the signaling network, which is essentially includes a number of genes you might have heard of, such as hedgehog, engrailed, patched, etc. There's a number of genes here, and these are the genes that form this network, and this network is the same in all the cells. All the cells across the embryo have the same gene network. However, they have different initial conditions because the mother cells that created them had different distribution of proteins in their cytoplasm. So even though the genes are the same in all these cells, their initial conditions are not. And because of that, and because of the specific network, certain patterns will form. And this, in fact, it's due to multistability, but I'm not going to talk about the multistability. That was something else. Other papers after this one talked a lot about that, but I just want to use the model as an example, and I'm describing it to you only with enough detail so that you can understand. So essentially, there's a number of cells next to each other, and at certain points, these lines, these dashed lines here, there's a pattern changes from one side of this dashed line to the other. And they represent in their model cells as hexagons, and they're basically side by side. And in this case, they actually, to not have spatial edge effects, they created and formed the whole thing into a toroidal structure. I'll talk a little bit more about the toroidal structure. Anyway, this model is very famous. It has many papers citing it. However, 2 years ago, I tried to access it, and I could not find it anywhere. There was no electronic version of this model available. In fact, after doing very, very, very deep searches, I did find an SPML model that contained an implementation of this. It was not by the authors, it was by other authors that produced it later on. But it was not a very satisfactory implementation because it didn't have reactions and species. It was just plain ODEs. So I decided to reconstruct this network myself. So I took on this task of encoding this model in COPASI. And just before I even go further, let me just say that had they constructed a network with square cells rather than hexagonal cells, we could actually already do this directly from COPASI. COPASI allows you to create arrays of compartments. So you start with one compartment, and then you say multiply this into a bigger array, and it copies all the reactions in that compartment to a larger number of compartments. However, they used hexagonal arrays, and hexagonal arrays COPASI cannot do. In fact, as I have to code this, I can tell you, if you're ever going to create a model like this, never use hexagons. Use squares, octagons, anything that is a power of a prime number. Do not use 6, which is not a power of a prime number. Essentially, you have to use 3 coordinates to do hexagonal arrays. They're not. You do not address them with 2 coordinates. You have to address them with 3 coordinates. So anyway, I had to develop code for that. So COPASI couldn't do it immediately. I mean, creating the model, COPASI can run the model easily, but I could not create it directly. I mean, but I would have to enter every single reaction of all the cells by hand, and I would not want to do that. So I then took this new library that my collaborator Frank Bergman from the University of Heidelberg created for COPASI. It's called Basico. It's essentially a Python API that allows you to write code in Python that essentially runs COPASI. And that's what I've done. I created a program that builds the hexagonal network, and they can reproduce the entire model that von Grasso et al. Had created. And as I said, otherwise, if it had been square, I could have done it very quickly, but it wasn't. So then I reproduced their model. It took a lot of work. Because it took a lot of work, I ended up writing a paper about it. This paper is actually about reproducibility, because I was testing how much do you have to report about a model in order to make it reproducible. And this was a perfect test because there was no model available. I had to redo it myself. So I had to get all the information from the paper and the supplementary data. And I still managed to reproduce it. I published this paper. I'm not going to go through the paper. You can read it if you want. It's freely available. However, that means I now have this model, which is where we're going to go. In fact, I not just created it, I also uploaded it to BioModel. So now the model is available for everyone. Anyone who wants to use this model, you can use at least the version with an array of 1x4 cells, which is the minimal needed to create the pattern. And they describe that in their original paper. So this is easy. As SPML versions available, COPASI versions available. You can run it in COPASI. You can run it in other software, as my paper describes, such as, for example, LibRoadrunner, etc., and any other software that runs SPML. So to give you a little bit more detail about the model, this is the diagram of one cell. This is the signaling network. The yellow boxes are mRNAs, and the green boxes are the proteins that they produce. So, for example, this is the engrailed protein, and this is the engrailed mRNA.
Mm-hmm.
And of course, the proteins generate, RNA generates proteins, and then the proteins also work each other. So, for example, this protein here can be cleaved by that one. And the cleaved version has certain activity that is different from the uncleaved version. And then some of these proteins are actually in the membrane. So these are the membrane proteins. These 4 different species are all membrane proteins. Some of them are receptors. This patched is a receptor. Another one, hedgehog, is actually a ligand. So hedgehog binds to the patched. This is the ligand, that's the receptor. So they happen on both cells, and this means that this cell communicates with that cell, because the hedgehog on the cell 01 binds patched on cell 02, and vice versa. Hedgehog on cell 02 can bind patched on cell 01. And this happens on all the edges of the hexagon. So in their model, the membrane is represented by 6 different compartments. So you have the 6 faces of the hexagon are different compartments, and these species that are on the membrane, they can diffuse to the nearer of the sides. So all of them can diffuse. The ones in this side can diffuse here or there, and then the ones in this side can diffuse there or here, and all along. So if there is a difference in concentration on one side, quickly that can diffuse out. And this is in one cell, and of course there's all the cells. Each of the cells is exactly like this, and they're all connected, and I'll show you in a moment the connections. I should mention this model with 4 cells, which is, as I said, the minimum required for the pattern to appear, has 152 chemical species, the entire model, the 4 cells, 480 reactions, which means 144 coupled ODEs. And this is not a problem for COPASI. This can be run in a few seconds. Mm-hmm. So how are the cells connected? As I said, it's a toroidal topology that they created to avoid edge effects. So in a 1x4 array, this cell 00 is connected to that cell 01, this side. So the molecules on this side, number 6, connect to the molecules on that side, number 3. But then also the molecules on side 4 connect with side 1 of the same cell, and all of them do that. But then also side 3 connects with side 6 on this side, side 2 connects with side 5 on this side 2 here connects with that side 5. This is basically a toroid, except that in this case it's only 4 cells. It's very hard to see them wrapping around themselves, but it is a toroidal approach. With a larger number of cells, it would be easier to visualize something like this. Okay? And so in this case, we're going to run the entire set of 4 cells with a single model in COPASI. And this is just to show you the results. I mean, my paper actually describes running this, so I'm not going to spend a lot of time. It's possible to do, and of course I did it, and it was published. So here are some results just to show you. These are 4 cells. I'm showing 2 proteins, the engrailed and the truncated version of Cubitus interruptus. And these are the patterns in the 4 cells in the array. And as you can see, there is a change in pattern here between the cells 1 and 2. They have more or less similar pattern. Also cell 3, but then cell 2 has a different pattern for engrailed. And in fact, it's kind of an opposite pattern for CN. CN is kind of higher here at the end time. This is getting close to steady state. And in this cell, it's actually suppressed. So for the 2 proteins, this cell is kind of different from the other 3. It's high here, it's low there. And this is the patterns that essentially will lead to the formation of the segment. So that's how it worked. So we ran it in COPASI, and basically that's it. Published paper, and I don't want to talk much more about this. I'm going to now move on to the other approach, which is what we wanted to do in this work. Okay, and so before I talk about the other approach, I need to introduce Vivarium, which is the software that my colleague Aaron Agmon has been developing for a number of years now. Mm-hmm. He started it at Stanford and is now here in Connecticut. And this is essentially an interface protocol. It's Python. It's a little bit more like a language. If you use Vivarium, you're going to have to write some code which represents how your agents behave. And it essentially allows to interface different simulation methods, and they don't have to necessarily be agent-based and ODEs. It could be ODEs and PDEs. It could be Boolean networks and agent-based. It could be flux balance analysis and ODEs, or a large number of these things connected. So his software is very, very general, very flexible, and it's composed of processes, stores, and composites. So the processes are essentially the subtypes of simulations. So each process has a number of, it's essentially like an agent, and it has a number of ports. These ports are essentially data that this process can communicate either to other processes or from other processes. They can be in one direction or bidirectional. These are basically the way in which this process can supply information or get information from other processes. Most of the variables will not be exposed because they're only needed internally. And so the other processes will not know how the internal state of this cell is. They will only know what is exposed by these ports. Then they also have stores. These are places where they can store information. It seems to be a simple concept, but it's ends up being very powerful in the way in this approach. And then they can make composites of these things into a so-called process bigraph, which is this here, where different processes are connected to each other eventually through stores. So these stores store numbers that are connected between the different processes. So the processes can read from each other through the stores. And then the simulation goes across, there is an orchestration. Vivarium basically orchestrates this, the Vivarium core it's called, where it basically does creates events, and those events are when it actually reads from processes and updates the store, or updates the processes from the store. And the processes don't have to be synchronous, they can be updated at different times. So in this case, we're showing two processes that are updated in asynchronous way, and Vivarium takes care of the state through that. So this is very, very generic, very abstract the way it is described. For those of you who are more from mathematically oriented or more computational, this should be very appealing. In fact, this was inspired by some of the work that Robin Milner carried out on bigraphs, which is explained, for example, very well in his book, which he wrote and was published, unfortunately, just a year before the author died. Also, there's a good review paper that he wrote in 2008 on these bigraphs. And this is all based on process algebra. It's based on pi calculus, I believe. And then what Eran did was to expand this to the so-called process bigraph. So he has a place bigraph and the process, a place graph and the process graph, and he combines them in this bigraph called a process bigraph, which includes these processes that are basically they're active things. They're not just stores of numbers. And he describes a little bit of this in his paper where he published the software a couple of years ago. So he has used Vivarium for the following things. One of them was to reproduce the work that the Covert Lab had published in 2020 on the E. coli whole cell model. This is a very large model of E. coli that represents 1,214 processes. The model has 19,000 parameter values, 10,000 mathematical equations. It couples different processes. As you can see here, this is the bigraph of the whole thing. Of course, we don't want to look at details, just a bit of zooming in a little bit where you can see metabolism is connected to a number of things, to the bulk. You get two-component signaling systems. You have some things that are in equilibrium. You have some molecular degradation, etc. You can produce curves of all sorts of things, like all the proteins, all the RNAs, all the tRNAs, etc. This is all aggregated from all of the processes. So his work was really was able to redo the entire software that they use in 2020, and in fact, it's actually rather more efficient. He then took that model of a whole E. coli and created a hierarchical thing. So with that one, he created many whole E. colis. So think of this as one E. coli cell with the full model with all the 19,000 processes 19,000 parameters in there. This is another E. coli. This is another one, another one, another one. And they're connected to an environment that includes diffusion of molecules and that also has a soft body physics simulator, so that the cells can bump into each other and not and basically take their space mechanically, push each other out. And so he inoculates a culture. This is one full E. coli model, and then they have division, and the culture grows, and all of these are different cells here. And then they use this to study heterogeneity in antibiotic responses. So just because of the stochasticity of gene expression, which is the model uses a stochastic simulation for the gene expression. And from that stochasticity, you end up finding out cells that end up developing resistance and others that don't. So you have this heterogeneity. I think here he's marking one protein that is what gives antibiotic resistance to the culture. Cells start dying out when you apply the antibiotic, but some cells survive because they have they express the proteins that allow them to be resistant. So this was done essentially. This approach, as you can see, it's a multiscale. It has a whole model, and then actually gets the entire whole cell model in each one of the agents. But again, also here, nobody studied what time frame should we use for updating cells between, right? So when should this each cell read about what happens to the other cells, neighboring its cells, and how often should it read of what is the concentration of the antibiotic in its environment? That has not been studied. They use some values, and as much as I understand, those values are fairly arbitrary. I suppose they studied them, but there's not a lot of description about it. And this is the same with most other multiscale models done until today. So we're going to study how this happens, and we're going to use the segment polarity network of Drosophila to do this. And we're going to use Vivarium and COPASI. And how we're going to do this is that the Vivarium approach is that we're going to code just one cell from the von Dassault model. And this cell basically just has the network that one cell has, and each one of them has that. And then Vivarium runs each one of the cells independently for a certain step size. And then we'll update all the concentrations across them. Vivarium does this at a certain amount, a certain time, which we can decide what that time is. And then they run again another step, for another certain amount of time independently. And then again, they get updated, and it continues going like this. So the only variable we're going to do here is change the step size. And then we're going to compare this model with the model that we already run, where we have all the equations coupled in COPASI, which we're going to treat as the ground truth. Now, of course, that model is also a model, right? So it's also an approximation. But we're asking, how much does the multiscale approximation deviate from the full set of differential equations? Now, because I told you that each cell is independent, I'm going to have to explain how they couple and how they are made independent. So before we make them independent, this is in the full model, the full ODE coupled model. We have this part. These are the proteins that matter, are these that are in the membranes. And I'm not showing here the one that diffuses across membranes. I'm only showing hedgehog binding to patched. But that happens in all the sides, in all contacts, in all cell-to-cell contacts. There's always hedgehog on one side and patched on the other. And this always happens, and this reaction has to go that way. And so these are the terms of the differential equation in the model. I'm highlighting here. These terms are from the neighboring cell, right? So I have the equation for cell 0, 1, but this term here is from cell 0, 2. And that term there and that term there. And of course, the same thing happens for all the other 5 cells neighboring this one, and for all the cells of all their neighbors. So how do we separate this? How do we make this cell independent? Well, we're going to do the following. We take away the next cell. We create one species here, which is an external species. It's external to this cell. It's whatever happens in the neighboring cell. And we're going to make that a constant. And it's a parameter of the model, at least for the time. If I can go back a couple of slides. During this time, those values are parameters of the model. However, they get updated at this time. So they are parameters for a certain amount of time, then they get updated, then they're parameters again. So if you think about it, throughout the whole time course, they're actually variables too. And in fact, if you update them very, very, very quickly, you would basically think that they were just variables. Okay, so that's how we do this. And Vivarium takes care of copying this value from this cell to the other one, and the other cell will have also similar value that then gets copied back here. So they get updated at that time. Yeah. So Vivarium also draws this topology map. This here is a map of the simulation. We have in these hexagons, which I marked in yellow, are the cells, and the circles are the connections between the cells. And of course, we only have to do half of them because they're bidirectional. So I'm only showing one half, but this connection is between this cell and that cell. It's from this side to that side. And of course, the one highlighted here is between this cell and that cell. And so it's between side 6 of this cell and side 3 of cell 01. And here, the molecules that are common between them are held constant, one for this side, one for that side, for that amount of time. And then when the Vivarium finishes the time step, it then exchanges the values. So they get to see, they update. So the next time step, this cell will now get some information from what happened in the previous cell. And of course, this happens to all of them. Whenever the software stops to update, it updates everything and updates everything simultaneously. At that moment, time is not passing. Okay, so we run this, and here is the time course. I don't know if you remember from the previous one, but it's pretty similar, as you can see. We run this with a time step of 2. We're running 200 units of time with a time step of 2 in this case. By the way, the time step that I'm talking about is the length at which the ODs go on their own. Internally, of course, they're being solved by an ODE solver, and they have whatever time step is required for having proper simulation. So they are actually run as the real time step is much smaller. But this is the time step of the agent-based approach. Okay, so here are some videos of what of the simulation looks like. I've highlighted a few molecules. So for example, here is the N-grailed and CN. Remember those two that have opposite patterns. This is the multiscale simulation. You could see the time at the beginning. So then, of course, they get into a steady state, which is the pattern that causes the segmentation. But then when this starts from 0, of course, that's how the concentrations go. These are concentration scales here, and the darker are higher concentrations, lighter, lower concentrations. So we're looking at those two molecules. Now you can look at other molecules. Now is this two. So this is the mRNA of Cubitus interruptus, and this is the active version of its protein. Nothing really special here. You see, protein goes a little slower than the mRNA. That's kind of normal in these type of things, because of course it needs to be expressed in this version, and then it needs to be cleaved. So it forms the pattern still. And then we can look at an mRNA and its corresponding gene. This is N-grailed, which is an important regulator here. So most of these genes are N-grailed is a transcription factor. So what it does, it just affects the production of Cubitus interruptus. And then it forms its own pattern. And then we can look at one of the membrane proteins. So now we're looking at hedgehog. We're looking at its mRNA, which is this top row. And then we look at the sum of the concentration of the 6 compartments. The mature version of this protein is in the membrane, right? So it gets produced and gets incorporated into the membrane. And we're looking at the concentration in the sum of the concentration in the 6 sides. So as you can see, it also works very much like actually the previous, the mRNA in its protein. So the simulation seems to work, and it produces results very, very similar to the ODE approach. I have a final one. This is patched, which forms the other pattern. So I need to move on. So here I'm actually plotting the two simulations, the ODE overlap, the multiscale overlap with the ODE. And it so happens you don't see the ODE because they was plotted first. So they are really overlapping. There is no measurable error that you can see. The approximation is pretty good. Now I should mention that this model, a very particular characteristic of this von Dassault model, is that they rescaled all the variables. So all the variables go between 0 and 1, which is a very interesting approach because it made the model completely non-stiff. There is almost no time scale in this model. Most molecules are in the similar time scale. As you saw, they were basically moving about the same dynamics. That's an interesting thing because it turns out in my paper, when I studied this in different software, some software had trouble because they tried to use stiff solvers to solve these equations, and the stiff solvers cannot solve these equations. You have to solve them with a non-stiff solver. COPASI uses one that is good for both, so we never see that problem in COPASI. Okay, so now we're going to study is changing. So I showed you the results with a step size that was good enough. It was a step size of 2. But how does the error grow? How does the error change with the step size change? This is exactly what we wanted to study. This is, I gave you all this introduction to give you the result. So we're now comparing, we're measuring the error between the ODE model and the Vivarium model. So one is the full coupled set of ODEs. The other one is the agent-based with ODEs approach. And we have two metrics here. One is the L infinity, which is basically the largest difference between one and the other. This is the maximum value of the difference of all the possible chemical species. We look at all of them through all the time points, and we look for which one has the highest difference. That's what we're plotting here. That's the so-called L infinity error, or distance. Then we're also plotting the root mean squared error, which is the square root. I forgot to put a square here. It's a square root of the sum. This should be squared, this term here inside the summation. So you sum all of the differences, divide them by the total number, and then you do the square root. This is very common. This is what people in protein structures use to characterize the structure of the crystals. And this is the RMS error. Now I put here a dashed line. This is the value we used. And as you saw, that was still pretty good error. We couldn't see differences visually between we can measure them, but we cannot see visually difference between the two simulations. Remember that the variables go from 0 to 1, so we're having errors of the order of 0.004, which is pretty low. They can go up. The maximum error can go up to 0.1. So 0.1 is already 10% of the range. So this would clearly be a bad result. You don't want to use a time step of 10. That's too large. But the time step of 2 was surprisingly efficient. Personally, I thought that we were going to have to go down to 0.1 or 0.01. But I was very surprised to find out that, and Iran was very happy to see that 2 actually worked well. Okay, so can we do this in larger models? So we moved on. We created a grid of 2 by 4. So remember, this is a toroid, but we can have more cells. Everything works this way. If you read the von Dassault paper, they show you also larger structures like this one, or even larger. So now we do this in COPASI again. We do the full ODE model. That's a 2 by 4 cell. Now has 304 species, 960 reactions, and 292 coupled ODEs. It works very quickly, as you'll see in a moment. I'll show you timings. It has more or less a similar error dependence. Actually, this one has a problem. I'll show you later on what the problem it is, but it's actually a bug. I don't believe that there is an issue, but you'll see it later on. So that worked pretty well. So we moved on further to 2 by 8. So now we have 2 by 8 cell model, 608 species, almost 2,000 reactions, nearly 600 coupled ODEs. Again, the error is very similar. A value of 2 still produces results that are very, very accurate enough that they can be, you can see no difference. So we will still want to do a little bit larger. One thing I should mention is the advantage of this model is, of course, of the Vivarium model, is that we can run each of these cells in a separate CPU. So this here was run on a machine with 16 CPUs, 16 cores. So each cell is run on its own time. As you will see, this is quite important for scaling. And in our high performance computing center, we can go up to 128 on the same machine. We could move across if we then use MPI, but we don't want to get there. Okay, so I mentioned before that the 2x4 cell had some problem. This is the RMS error of all three models, the 1x4, the 2x4, and the 2x8. The 1x4 and 2x8 are both here overlapping. You don't see any difference. They have exactly the same RMS error dependency on the step size. This 2x4 has much larger error, and in fact the error is more or less fixed across this range. And I'm absolutely sure this is an artifact of the way I did my not the way I did the computations, but the way the files were stored and copied across machines. And I spent this afternoon trying to fix it, but I had to give you the talk today, and I could have just hidden this model, but I decided that maybe at the time I'll show you everything. Whenever we get to publish this, I will have fixed this. I'm pretty sure it's an artifact. I do not believe this is an issue in the model, because as you can see, both the 1x4 and the 2x8 have exactly the same error. So why would the 2x4 have a different error? There's no reason. Okay, so how about runtime? And I'm just getting very close. This is one of the last slides, last slide of results. So how about the run times? So how long does it take to run each of these models? So I'm plotting here these three lines that go down are the multiscale model, and this is the ODE model. Now you don't see the ODE model changing because the ODE model has variable step size. And so it doesn't matter what interval you run here, it's always the same step size. The step size is much smaller than the 2 seconds that we output the data. So that's why it takes the same amount of time to get values every 10 seconds or values every 0.01 second or values every millisecond. The simulation has a much smaller step size than that, so it's always the same amount of time. And it's about less than 1 second for the 1x4 model. So COPASI run those 100 and something equations in less than a second, solve them all. The 2x4, it's a little bit more. It's maybe, it says logarithmic, right? So it's about 2 seconds. And then, so the timing is actually increasing more or less linearly. But then it's still about 3 seconds to run the 2x8. So now you ask, why would you want to do the multiscale if the OD is so fast? Well, a couple of reasons. I mentioned them before. You cannot use the ODE if the cells are moving around, or if new cells appear and old cells disappear. So this example, we used especially to do the comparison, the cells are fixed. And so we can show this. But of course, if the cells were moving, we could not do this. It would be impossible to do this. So the other approach would have to be done. And now we know that at least potentially this kind of time scale is okay for the timings, the location. So we use 2 seconds, which is here. And you can see at 2 seconds, it's more or less where the lines cross. So it turns out that the multiscale simulation is not that much slower. In fact, for the 2x4, it's about the same time, running it multiscale or running the full ODE. Now, this is because we can parallelize. This is running with 16 CPUs, whereas COPASI is only using 1 CPU. That's why the two are more or less the same time. But of course, if I could make the time even larger, if the error was good enough to make a value of 10, then I could get much larger, much faster. Of course, if I have more cells, as you could see, then most likely we would be faster, because see the crossing here, crossing for the 2x8 is here, crossing the 2x4 is there. So the larger the model, the earlier is going to be the crossing. So with more cells, the multiscale gets advantage. So there's some take homes from here. One, the scaling is very good on the multiscale approach with Vivarium. We're very happy with this. And, until you have enough cores to throw at the problem, you will speed it up. The other thing is that 588 coupled ODEs are still not that slow to calculate. This is, do not underestimate the ODE approach. It's still pretty powerful. Okay, so just to summarize, I think this is the first systematic study of approximation error in multiscale agent-based ODE approaches, coupled agent-based and ODE simulations. I cannot say for sure. I'm still trying to look in the literature because I somehow I think that maybe people should have looked at this before, but so far I have not found anything. In this model that I remind you is non-stiff, the multiscale approximation can really take very large time steps and therefore makes it competitive with using with coupled ODEs. However, we don't know if this happens in stiff models. Probably not. Scaling is optimal while the number of cores can match the number of cells. Of course, this is not going to work out for millions of cells, but it's going to be fine if you have hundreds of cells. And so this is our conclusions from this little study so far, what we want to do further. And one of the reasons why this has not or is not going to be published right away is we want to study other models, particularly ones that are stiff, because of course stiffness is going to affect how soon we have to communicate. Potentially, I think. Intuitively, I would think so. So we still want to do that. And of course, then we will publish.
Yes.
I'd just like to thank a few people. Apart from Aaron Agmon, who is a co-author, so I'm not going to thank him. He's a partner in crime. But Frank Bergman was really, really helpful. When we started using Basico, I mean, he's still building Basico and he's adding features when they're needed. We needed a few that he added, overnight. We asked him, how can we do this in COPASI? We could do that, but in Basico we couldn't. So he immediately provided it. Next day or so, he would have code in GitHub available. Also, he managed to speed up his code by 2 orders of magnitude. So I hope that he will also thank us, because we found out some bottlenecks that his code had, and because of our simulations, he managed to speed it up. Joe Madison is my PhD student. It's actually an MD PhD student, and he's now working on the medical part. He actually was the first person that used Vivarium with COPASI. He was modeling a crypt-villus system for enterocytes in the intestine, in the small intestine. And then a ex-postdoc of mine, Joseph Dada, many years ago, we both worked on a software which we never ended up really publishing, called ManyCell, which was a multiscale simulation of this approach, which we used to simulate growing cultures of Saccharomyces cerevisiae. However, we really never published. I mean, we published only in a conference paper. He's now in Nigeria. He's a professor there. He's an electrical engineer. He's working in electrical engineering. So that code was stagnated. And that's why I'm doing this now, because, I had a little bug already to try and do this. And my colleagues that are also authors of COPASI, and the National Institutes of Health for funding our work. And so I'm happy to answer any questions that you may have.
Okay, thank you.
Thank you.
Pedro. Okay, let me stop the recording.